In the silent, microscopic world within us, a war is being fought every second of every day. It is a war of survival, waged by a vast and deeply complex defense network, the immune system. This system is the sole agency tasked with protecting the nation state of the body from a relentless tide of foreign invaders and internal traitors. When it operates flawlessly, we live in blissful ignorance of the constant battles fought on our behalf. But when its protocols fail, when its agents are compromised, or when its very weapons are turned against the citizens it's sworn to protect, the consequences are catastrophic. This is the story of that agency, a chronicle of its triumphs and its most devastating failures. We will investigate the cases where it was too weak, allowing deadly infiltrators to overrun the system, and the cases where it was too strong, leading to violent, self-destructive civil wars. To understand these conspiracies of biology, we must first be briefed on the agency itself, its structure, its operatives, and its rules of engagement. The agency operates in two distinct, yet interconnected, divisions. The first is the division of innate immunity. Think of these as the first responders, the uniformed officers always on patrol. They are ancient, hardwired, and brutally efficient. Their response is immediate. They are programmed to recognize the common signatures of hostile entities and the distress signals of cellular damage, launching a swift, uncompromising counterattack. They are the first line of defense, designed to contain a threat the moment it appears. The second, more clandestine division is adaptive immunity. These are the elite specialists, the intelligence analysts, and covert operatives. They are not pre-programmed for every threat. They must be activated. They must learn the identity of a specific enemy, gathering intelligence and developing a highly specialized and devastatingly powerful plan of attack. Their response is slower, but it is precise, powerful, and most importantly, it is remembered. Should the same enemy ever dare to return, the adaptive division will be waiting, ready to neutralize it with overwhelming force and speed. The file on that threat is never closed. Our investigation begins with the first responders, the innate division, the ever-present guardians. The first line of defense is not a cell, but a structure, the epithelial barriers. These are the great border walls of the body, the skin, the lining of the gut, and the airways. But these are not passive fortifications. They are an active security perimeter, patrolled by their own lymphocytes and armed with chemical weapons like antimicrobial defensins, ready to neutralize threats at the point of entry. But walls can be breached. When an infiltrator makes it past the perimeter, the agency's cellular operatives are deployed. The first on the scene are the phagocytes, the frontline soldiers. Neutrophils are the shock troops, rapidly recruited from the blood to sites of invasion. Monocytes circulate as reserves, but when they march into the tissues, they transform into the formidable macrophages, larger, longer-lived operatives that not only destroy invaders, but also clean up the battlefield of cellular debris. Specialized garrisons of these macrophages stand permanent guard in critical territories, the Kupffer cells in the liver, the microglia in the brain, and the alveolar macrophages in the lungs. Their directive is simple: identify, engulf, and destroy. While macrophages are the heavy infantry, the dendritic cells are the field intelligence agents. They are stationed as sentinels in the tissues, where their primary mission is not to kill, but to gather intel. They capture fragments of the enemy antigens, and their directive is to race from the front lines to the agency's command centers, the lymphoid organs. There, they present this critical intelligence to the elite adaptive division, initiating a far more sophisticated counter-espionage operation. Alongside them work the innate lymphoid cells, or ILCs, including a particularly deadly operative, the natural killer cell. The NK cell is the agency's internal affairs assassin, tasked with an incredibly sensitive mission, identifying and eliminating traitors from within. They patrol our tissues, hunting for our own cells that have been compromised by viruses or have begun the malignant transformation into cancer. And finally, there is the agency's heavy ordnance, the soluble proteins of the blood. Chief among them is the complement system, a network of plasma proteins that, when triggered, unleashes a cascading chain reaction. It can coat invaders in molecular flags that mark them for destruction, or it can assemble itself into a deadly weapon that punches holes directly into the membranes of microbes, causing them to burst. But this raises the most critical question of all. In a world of trillions of cells, with friend and foe intermingling, how does this agency, with its arsenal of brutal weapons and lethal operatives, know who to kill and who to protect? We have now concluded our initial briefing on the components of the innate immune system, the first responders. In our next transmission, we will investigate their surveillance technology, the network of receptors they use to distinguish a hostile entity from a loyal citizen, a critical step before the system-wide alarm is sounded for the elite adaptive forces. The innate system's operatives are fast and lethal, but their actions are not random. They are governed by an intricate surveillance grid, a network of receptors designed to solve the most fundamental problem in espionage, distinguishing friend from foe. This system does not look for individual identities. Instead, it is programmed to recognize broad, unmistakable signatures of danger. It looks for pathogen-associated molecular patterns, the recurring molecular structures that act like the common uniform of microbial armies, features they cannot easily change without compromising their own survival. It also detects damage-associated molecular patterns, the internal distress signals released by our own dying or injured cells, the biological equivalent of a scream for help. The receptors that form this grid are called pattern recognition receptors. The most famous are the toll-like receptors, or TLRs, a family of scanners positioned on the cell's outer membrane and within its internal compartments. When a TLR detects its target, a piece of a bacterium, a strand of viral RNA, it trips an alarm. This alarm activates master protocols deep within the cell. One is NF-kappa B, the call to arms signal, which triggers a firestorm of inflammation by ordering the production of cytokines and recruiting more agents to the scene. The other is the interferon regulatory factors, a protocol that initiates a system-wide antiviral lockdown, stimulating the production of cytokines that can halt viral replication in its tracks. But what about threats that have already breached the outer defenses and are loose inside the cell? For this, the agency deploys internal sensors, the NOD-like receptors, or NLRS. These cytosolic detectors are the last line of defense, sensing a stunning array of internal threats, from microbial components to the toxic byproducts of cellular damage like uric acid crystals. When these internal sensors are tripped by a sign of catastrophic failure, they assemble a multiprotein complex known as the inflammasome. Think of it as a biological dead man's switch. Its assembly activates an enzyme that unleashes a powerful cytokine, interleukin-1, a potent flare signal that ignites a massive inflammatory response, recruiting reinforcements and causing fever. This very mechanism, when it mistakenly identifies uric acid crystals as a threat, is the cause of the excruciating inflammation in gout. This innate division is a masterpiece of immediate, brutal efficiency. But it is a blunt instrument. It has no memory. It fights every battle like it's the first time. Its most critical, strategic function is to contain the initial breach and, crucially, to sound the alarm that activates the agency's most sophisticated and powerful weapon, the adaptive immune system. The adaptive division. Here, the rules of engagement change entirely. This is the world of specific, targeted operations, of learning and memory. The adaptive division has two primary branches of operation. Humoral immunity is its long-range weapons program, run by B lymphocytes, who develop and deploy antibodies, molecular smart missiles that can seek out and neutralize threats in the open fluids of the body. Cell-mediated immunity is its special operations force, run by T lymphocytes, designed to hunt down and eliminate enemies that have already infiltrated our own cells, hiding from the antibody missiles. The elite agents of this division are the lymphocytes. They are not stationed in one place, they are constantly mobile, circulating through blood and tissue in a ceaseless surveillance mission. An agent who has not yet faced a real threat is considered naive. Once activated by a specific enemy, it becomes an effector cell, an operative on a live mission. and after the mission is over, a few remain as memory cells veterans who hold the permanent file on that enemy, ensuring that any future encounter will be met with a swift and overwhelming response. The activation protocol for these agents is one of biology's greatest marvels: clonal selection. The agency maintains billions of different lymphocyte clones, each with a unique, randomly generated receptor unique key. When a foreign antigen lock is detected, the system doesn't waste time training a new agent. It searches for the one agent in a billion who already possesses the perfect key. Once found, that single agent is selected and ordered to clone itself into a massive army, all bearing the identical key needed to neutralize that specific threat. The operatives of the T-cell branch are the master coordinators and assassins. A T-cell's scanner, its T-cell receptor TCR, is incredibly specific, but it has a critical limitation: it cannot see a raw threat. It can only recognize intelligence, a small peptide fragment of an enemy, after it has been processed and displayed on an official agency terminal, the MHC molecule. The type of terminal determines which agent can read the intel. MHC Class I terminals are standard issue on nearly every cell in the body. They constantly display samples of proteins from inside the cell, a status report to the patrols passing by. If a cell is compromised by a virus or cancer, it will display a foreign peptide on its Class I terminal. This is a distress signal that can only be read by a CD8 plus cytotoxic T-cell, the killer. Upon reading this signal, the CD8 agent's directive is clear and final: terminate the compromised cell. MHC Class Roman II terminals, however, are restricted hardware, used only by professional intelligence-gathering cells like dendritic cells. They display intel on threats that have been captured and ingested from the outside world. This intel can only be read by a CD4 plus helper T-cell, the commander. The CD4 agent doesn't carry out the kill itself. Its mission is far more critical: it analyzes the threat and then coordinates the entire adaptive operation, issuing cytokine commands that activate the CD8 killers, energize macrophages, and, crucially, grant clearance to the B-cell division to begin its weapons program. When a B lymphocyte, the weapons developer, gets that clearance, it transforms into a plasma cell, a single-minded factory that can churn out thousands of antibody missiles per second, designed to hunt down the specific extracellular enemy that triggered the alarm. We have now concluded the briefing on the agency's fundamental structure and its key operatives. We understand the protocols of the innate first responders and the elite specialists of the adaptive division. In the next part of our investigation, Part 2, Friendly Fire and False Alarms, we will open the case files where this powerful and intricate system malfunctions, leading to the destructive chaos of hypersensitivity. An effective defense agency must be powerful, but it must also be disciplined. The greatest threat to any nation is not always the enemy at the gates, but the risk of its own weapons being turned against its people. We now enter the dark side of immunity, the case files where the system's awesome power is unleashed against the wrong targets, at the wrong time, and with the wrong intensity. These are the chronicles of hypersensitivity. These pathological protocols are not born from new or alien weapons. They are the agency's standard operating procedures, simply misdirected. The damage is caused by the very same soldiers and weapons designed to protect us, now deployed in a chaotic and self-destructive manner. The triggers for these malfunctions are varied. Sometimes it is an overreaction to a harmless foreign entity, a false alarm that spirals into the full-blown crisis of allergy. At other times, and most tragically, the system targets its own loyal cells, initiating a devastating civil war known as autoimmunity. Our investigation has identified 4 distinct protocols for this self-inflicted damage, 4 ways the agency can betray its mission. Case File 1, Type I: Hypersensitivity, the false alarm. This is the story of a catastrophic intelligence failure. The system misidentifies a harmless intruder, a speck of pollen, a protein from a peanut, as a mortal threat, and launches a response of shocking, disproportionate violence. The plot begins with a first quiet encounter. The allergen is captured and presented, but instead of being dismissed, it triggers a specific class of operatives, the Tro helper cells. These cells issue a bizarre and fateful directive to the B-cell division, mass produce a specialized class of antibody, IgE, designed to target this non-threat. This IgE does not patrol the blood. Instead, it docks onto the surface of mast cells, the agency's granular sentinels stationed in tissues throughout the body. The mast cells are now armed. They have been turned into sleeper mines, each one primed by IgE and waiting for the enemy that exists only in the system's flawed intelligence. When the second exposure occurs, the trap is sprung. The allergen drifts into the tissue and cross-links the IgE molecules on the surface of a mast cell. The detonation is instantaneous. The mast cell degranulates, violently expelling its preloaded chemical arsenal. Chief among these is histamine, a weapon that causes immediate chaos. Blood vessels are ordered to dilate and leak fluid, causing massive swelling. Smooth muscles are commanded to contract, constricting the airways. This is the immediate reaction, the sudden, violent assault responsible for the familiar miseries of allergy and the terrifying, life-threatening bronchospasm of an asthma attack. But the danger is not over. The mast cell's detonation also releases signals that call for reinforcements, setting the stage for a prolonged siege known as the late-phase reaction. Hours later, a second wave of operatives, primarily the highly destructive eosinophils, arrive at the scene. Attracted by the chemical signals, they release their own toxic proteins, causing further tissue damage long after the initial false alarm. This two-act drama can be a localized nuisance, like hay fever, or it can be a systemic catastrophe. If the allergen enters the bloodstream, it can trigger mast cells throughout the body simultaneously, causing systemic anaphylaxis, a state of widespread vascular collapse and shock that can kill in minutes. Case File 2, Type Roman II hypersensitivity, the targeted assassination. In this protocol, the betrayal is far more direct. The agency's own long-range weapons, IgG and IgM antibodies, are programmed with a kill order against targets located on the surface of our own cells. This is not a false alarm, it is a targeted assassination, carried out in one of three ways. First, marked for death. An antibody directly tags a loyal cell, such as a red blood cell or a platelet, with what is effectively a kill tag. This process, called opsonization, marks the cell for immediate execution by the agency's phagocytic cleanup crews. This is the central plot of autoimmune hemolytic anemia, where the body systematically destroys its own blood supply. Second, inflammatory collateral damage. Here, antibodies bind to fixed targets within a tissue, like the basement membrane of a kidney. This deposition acts as a beacon, activating the complement system's demolition cascade and summoning legions of neutrophils to the site. The neutrophils arrive, but they cannot eliminate the target. In their frustration, they do the only thing they are programmed to do: they unleash their entire arsenal of destructive enzymes, bombarding the healthy tissue and causing massive collateral damage. The third and most insidious method is sabotage. The antibody does not kill the cell, it hijacks its function. It can act as a blocker, binding to a critical receptor and preventing it from receiving its intended signal, the mechanism that causes the profound muscle weakness of myasthenia gravis. Or it can do the opposite, acting as a rogue signal that jams a receptor in the on position, forcing a cell into a state of runaway activation, as seen in the hyperthyroidism of Graves' disease. Case File 3, Type Roman III hypersensitivity, the unseen trap. This case is different. The damage is not caused by a direct attack, but by the formation of immune complexes, rogue convoys of antigens and antibodies floating freely in the bloodstream. These complexes are too small to be cleared efficiently, but large enough to get stuck, lodging themselves in the body's fine filters, the delicate blood vessels of the kidneys, the joints, and the skin. Once trapped, these complexes trigger the complement system, which sends out a distress signal. Neutrophils rush to the scene, but they find an enemy they cannot engulf. Frustrated, they unleash their corrosive enzymes directly onto the vessel wall, causing vasculitis, a destructive inflammation that damages the very pipelines of the body. This is the central mechanism of injury in systemic lupus erythematosus. Case File 4. Type Roman 4 Hypersensitivity, the sleeper cell activation. Our final file details an operation run entirely by T cells. There are no antibodies. The response is slow, deliberate, and often far more destructive. It is a delayed type hypersensitivity. The first protocol is incitement to riot. CD4 plus helper T cells, having been previously sensitized to an antigen, are reactivated. They release a flood of cytokine signals that act like propaganda, whipping local macrophages into an inflammatory frenzy. This sustained, macrophage-driven assault is responsible for the chronic tissue damage and granuloma formation seen in diseases like tuberculosis. The second protocol is a direct hit. CD8 plus cytotoxic T cells, the agency's professional assassins, are given a kill order for a specific self-antigen being displayed by our own cells. They then systematically hunt down and execute every cell bearing that protein, leading to the targeted destruction of an entire tissue, such as the assassination of the insulin-producing beta cells of the pancreas in type 1 diabetes. We have now examined the 4 primary protocols for friendly fire and false alarms within the immune agency. We've seen how its weapons can be turned against us through explosive allergic reactions, targeted assassinations, collateral damage, and direct T-cell insurrections. This leads to the most chilling investigation of all. What happens when the target of these pathological protocols is not an external substance, but the very essence of the self? In our next briefing, we will delve into Part 3, the Civil War and the profound betrayal of autoimmune diseases. For every defense agency, there is one nightmare scenario, one threat that looms larger than any foreign adversary. It is the threat of internal collapse, of a civil war where its own operatives, armed with its most powerful weapons, turn against the very citizens they are sworn to protect. This is the world of autoimmunity. It is not simply a malfunction, it is a fundamental breakdown of the system's most sacred protocol, the ability to distinguish self from non-self. It is a war waged not at the borders, but in the heartland of the body, in the joints, the kidneys, the skin, and the brain. And it is a war with a troubling and mysterious connection to our modern world, with an incidence that is steadily rising. To understand how such a catastrophic betrayal can occur, we must first examine the sophisticated fail-safe systems designed to prevent it. We must investigate the agency's loyalty conditioning program, a set of protocols known as immunologic tolerance. This is the process that ensures the billions of agents in our immune system can coexist peacefully with the trillions of loyal cells that make up our body. This loyalty programming occurs in 2 phases. The first is central tolerance, and it takes place at the agency's training academies, the bone marrow for B-cell cadets and the thymus for T-cell cadets. Here, immature lymphocytes are rigorously tested before they are ever allowed to enter active service. As they develop, their newly formed antigen receptors are exposed to a vast library of the body's own self-antigens. Any cadet whose receptor binds too strongly to a self-protein is immediately flagged as a potential traitor, a threat to internal security. The protocol for these cadets is swift and decisive. They are eliminated by apoptosis, a clean and efficient process of programmed suicide known as clonal deletion. In the thymus, a master regulator gene called AIR acts as a simulator, forcing the expression of proteins normally found only in peripheral tissues, ensuring that T-cell cadets are tested against the widest possible range of self-targets. For B-cell cadets, there is sometimes a final chance at redemption, a protocol called receptor editing, where the cell is given an opportunity to reshuffle its receptor genes to create a new one that is no longer self-reactive. If it fails, termination is the only outcome. But no training program is perfect. Some potentially self-reactive agents inevitably escape the academies and make it out into the field. This is where the second phase of security comes into play, peripheral tolerance. These are the protocols designed to disarm or eliminate rogue agents in the peripheral tissues before they can cause harm. One such protocol is anergy, a force stand down. An agent's activation requires two signals: recognition of its target and a simultaneous confirmation signal from a professional antigen-presenting cell. If a self-reactive T cell in the periphery finds its self-antigen on a normal tissue cell that lacks the authority to give this second signal, the T cell is not activated. Instead, it enters a state of permanent unresponsiveness. It is disarmed and rendered inert. If anergy fails, the system deploys its own internal police force, the regulatory T cells or TREGs. These are elite operatives programmed by the master gene FOXP3, whose sole mission is to maintain order. They actively patrol the body, hunting down and suppressing overactivated or self-reactive T cells, preventing them from launching unsanctioned attacks and keeping the peace. Finally, there is a protocol for field termination. Some mature lymphocytes that repeatedly recognize self-antigens are programmed to engage a self-destruct sequence involving a death receptor known as FAS. This engagement triggers apoptosis, eliminating the dangerous agent from the field. With so many layers of security, the loyalty of the agency seems assured. So we are left with the central mystery: how do these fail-safes fail? The evidence suggests that autoimmunity is not caused by a single catastrophic event, but by a conspiracy between two factors: genetic susceptibility, the insider vulnerabilities, and environmental triggers, the external catalysts that ignite the conflict. The genetic investigation consistently points to a prime suspect: the family of HLA genes. These genes code for the MHC molecules, the very terminals that display intelligence to T cells. Certain variants of these HLA molecules seem to be inherently predisposed to improperly displaying self-peptides in a way that makes them look foreign, creating a critical vulnerability in the system's core identification protocol. Other genes, like PTP-N22, are implicated as well, suggesting defects in the de-escalation pathways that should normally prevent operatives from overreacting. But a genetic vulnerability is not enough. An external trigger is almost always required to light the fuse. The most common and most studied of these triggers are infections. An invading microbe can create a war zone of intense inflammation. In the chaos of this battle, with alarm signals firing everywhere, dormant self-reactive T cell sleeper agents that should have remained quiet can be accidentally activated by the widespread distress signals, a phenomenon known as bystander activation. Even more insidiously, an infection can trigger autoimmunity through molecular mimicry. This is a classic espionage plot. An invading microbe possesses proteins that bear a striking resemblance to our own. The immune agency mounts a powerful and appropriate response against the foreign invader. But the weapons it creates, the antibodies and T cells, are now programmed to recognize a target that looks tragically similar to a loyal self-protein. The agency, in its fight against the enemy, inadvertently creates a weapon that will now hunt its own citizens. This is the mechanism believed to cause the heart damage in rheumatic fever, where the response to a streptococcal infection cross-reacts with proteins in the human heart. We have now examined the agency's most critical protocols, the loyalty tests designed to maintain self-tolerance, and we've identified the prime suspects in their failure: a toxic combination of insider genetic vulnerabilities and external triggers, like infections. The stage is set for disaster. In our next transmission, we will open the first and most enigmatic case file in this civil war, the multi-system conspiracy known as systemic lupus erythematosus. Case file: Systemic lupus erythematosus SLE. SLE is the great imitator, a phantom menace. It is a chronic, relapsing, multi-system autoimmune disease, a conspiracy so widespread it can strike any organ in the body. The typical patient is a young woman presenting with a mysterious constellation of symptoms: a telltale butterfly rash across her face, fever, debilitating joint pain, and signs of kidney distress. The primary evidence in this investigation is the astonishing array of illegal weapons found in the patient's system, autoantibodies. The single most important finding, present in virtually every case, is the anti-nuclear antibody, or ANA. This is the smoking gun. It is an antibody inexplicably designed to attack the most secure and vital command center in every cell, the nucleus. Its presence signifies a profound and systemic betrayal. Forensic analysis of these ANAs reveals different ballistic signatures, hinting at which nuclear components are the primary targets: DNA, histones, or other core proteins. Two of these are so specific they are considered a calling card for the disease: antibodies to double-stranded DNA and to the Smith antigen. The investigation into SLE reveals a dark and self-sustaining conspiracy, a vicious cycle that amplifies the betrayal. It begins when an environmental trigger, most often ultraviolet radiation from sunlight, acts as an accomplice. UV light causes damage and apoptosis in skin cells, forcing them to unmask their nuclear contents. In a healthy individual, this cellular debris would be cleaned up swiftly. But in patients with SLE, a defect in this cleanup operation, often linked to genetic deficiencies in the complement system, leaves this nuclear evidence scattered across the crime scene. This is where the genetic vulnerability and self-tolerance becomes critical. The patient's rogue B cells and T cells see these exposed nuclear antigens not as debris, but as a hostile threat. They begin to produce the first wave of anti-nuclear antibodies. These antibodies then bind to the nuclear antigens, forming immune complexes. And here, the conspiracy turns inward and begins to feed itself. These newly formed immune complexes are seen as an even greater threat. They are ingested by the patient's own B cells and dendritic cells. Once inside, the nuclear DNA and RNA from the complexes trick the internal antiviral alarm systems, the toll-like receptors. This is a catastrophic error. The system is tricked into believing it is under a massive viral attack. In response, it unleashes a flood of type I interferon's powerful cytokines that escalate the entire immune response to a state of high alert. This, in turn, drives the B cells to produce even more higher affinity anti-nuclear antibodies. The result is a terrifying feedback loop. The response creates more immune complexes, which trigger more interferon, which drives an even bigger response. It is a biological firestorm, perpetually fueling itself. The damage left in the wake of this firestorm is widespread and severe. The primary mechanism of injury is the one we investigated earlier: type Roman-3 hypersensitivity. The massive burden of circulating immune complexes deposits in the fine filters of the body, the small blood vessels of the kidneys, the skin, and the joints, where they trigger a destructive inflammatory assault. Additionally, other autoantibodies wage a more direct war via type Roman-2 hypersensitivity, targeting blood cells for destruction and leading to anemia and low platelet counts. We have now uncovered the conspiracy at the heart of systemic lupus erythematosus, a self-sustaining cycle of autoantibody production initiated by a failure to clear cellular debris and pathologically amplified by the body's own antiviral alarm systems. In our next transmission, we will survey the crime scenes left in the wake of this attack, examining the widespread damage in the kidneys, skin, and joints, before moving on to other chilling cases of autoimmune betrayal. The damage report for systemic lupus erythematosus is a grim testament to the scope of its internal war. The primary evidence of the attack is found in the body's smallest blood vessels. Here, the deposition of immune complexes triggers an acute necrotizing vasculitis, leaving behind a smudgy, eosinophilic scarring known as fibrinoid necrosis, the scorched earth of a microscopic battle. Nowhere is this damage more consequential than in the kidneys, the primary target in more than half of all cases. Investigators have classified the assault into several stages of escalating severity. It begins as a quiet infiltration, with immune complexes depositing silently in the mesangium. But it can rapidly escalate to a full-blown siege, a diffuse proliferative lupus nephritis. Glomeruli, the kidney's delicate filtering units, become battlegrounds, swollen with proliferating cells and inflammatory infiltrates. Under the microscope, investigators find the telltale evidence: thick, wire loop-like deposits of immune complexes trapped beneath the endothelium, a sign of a relentless and overwhelming attack. As the battle rages, the filters are progressively destroyed and scarred, leading ultimately to renal failure, one of the most common causes of death in this disease. On the skin, the conflict is often tragically visible. The characteristic butterfly rash is the outward sign of the war within. Microscopically, the dermoepidermal junction is a zone of liquefactive degeneration, a trench where the basal layer of the epidermis is being systematically destroyed, with immune complexes and complement proteins lining the area like unexploded ordnance. The conspiracy is truly systemic. In the joints, it manifests as a painful synovitis. In the heart, it can lead to Liebman-Sachs endocarditis, where distinctive small, warty vegetations appear on the heart valves. In the spleen, the arteries can become wrapped in concentric layers of fibrous tissue, creating bizarre onion skin lesions. The evidence is clear: no organ is safe. Case file: Sjögren syndrome, the drought. From the widespread, explosive chaos of lupus, we turn to a more focused and insidious attack. The case of Sjögren syndrome is not a multi-front war, but a targeted act of sabotage against a specific piece of the body's infrastructure, the moisture-producing lacrimal and salivary glands. The clinical presentation is a mysterious and debilitating drought. The patient suffers from dry eyes, known as keratoconjunctivitis sicca, and a dry mouth, or xerostomia. The underlying cause is a stealthy infiltration of the glands by a combination of CD4+ helper T cells and B cells. These rogue operatives are believed to be reacting to an unknown self-antigen within the glandular tissue, or perhaps to a viral trigger that initiated the betrayal. Over time, this lymphocytic infiltrate systematically destroys the glandular architecture, leading to atrophy and fibrosis. The flow of tears and saliva slows and then ceases. While the attack is focused, investigators find serologic clues that hint at a wider conspiracy: the presence of telltale autoantibodies, specifically those targeting the ribonucleoprotein antigens SSRO and SSB-LAG, which act as markers for the disease. Patients with Sjögren syndrome are also at a disturbingly high risk, a 40-fold greater chance than normal of the conflict escalating into a full-blown B-cell lymphoma, a malignancy born from the chronic, unceasing inflammation within the glands. Case file: Systemic sclerosis, the suffocation. Perhaps the most relentless autoimmune attack is not one of explosive inflammation, but of a slow, progressive suffocation. This is the case of systemic sclerosis, or scleroderma. The hallmark of this disease is a pathological excess of fibrosis, a runaway scarring process that hardens the skin and chokes the life from internal organs. The investigation into its cause points to a conspiracy between three processes. First, an autoimmune response, where activated CD4+ T cells release cytokines like TGF-beta and IL-13 that act as relentless commands, ordering fibroblasts to produce more and more collagen. Second, widespread vascular damage. The body's smallest blood vessels become damaged and narrowed, leading to chronic ischemia, which in turn triggers even more fibrosis as a form of dysfunctional repair. Finally, the fibroblasts themselves appear to be intrinsically abnormal, hyperreactive to the fibrogenic signals. The result is a slow, creeping petrification. The skin of the fingers, hands, and face becomes tight and bound down. The face can become a drawn, immobile mask, and the fingers can curl into a claw-like deformity. Internally, the esophagus loses its flexibility, turning into a rigid tube that severely impairs swallowing. The most ominous developments occur in the lungs, where fibrosis can destroy the delicate architecture of the alveoli, and in the kidneys, where vascular damage can trigger malignant hypertension and renal failure. Forensic serology provides critical clues. The presence of an antibody to DNA topoisomerase I is linked to the diffuse, aggressive form of the disease. In contrast, an anticentromer antibody is often found in patients with a more limited version of the disease, sometimes called the CREST syndrome, a variant with a more insidious and prolonged course. We have now closed the major case files on the autoimmune civil war, from the systemic chaos of lupus to the targeted sabotage of Sjögren's and the relentless fibrosis of scleroderma. The agency's capacity for self-destruction is terrifyingly clear. Our investigation must now turn to a different kind of threat, one that challenges the agency's core directive in a unique way. In our next briefing, Part 4, The Foreign Invasion, we will analyze the agency's response to a deliberately introduced foreign entity, a transplanted organ. The immune system is a master of border control, ruthlessly programmed with a single, unyielding directive: identify and eliminate anything that is non-self. This raises a profound challenge. What happens when a foreign entity, a life-saving organ from another individual, is deliberately introduced behind our own lines? The result is one of immunology's most dramatic conflicts. The agency, doing precisely the job it was designed to do, recognizes the allograft as a massive, sophisticated invader and mounts a full-scale operation to destroy it. The primary foreign signature that triggers the alarm is the set of HLA molecules on the donor organ's cells. Because these are inherited genes, they are unique to every individual, acting as a molecular fingerprint. To the recipient's immune agency, the donor's HLA fingerprint is an unmistakable sign of a foreign invasion. The interrogation of this threat proceeds down two pathways. The direct pathway is an immediate confrontation. The foreign organ arrives with its own embedded passenger cells, the donor's antigen-presenting cells. The recipient's T cells directly interrogate these foreign cells, recognize their HLA terminals as hostile, and instantly sound the alarm. It is a swift and powerful trigger for an attack. The indirect pathway is a more classic intelligence-gathering operation. The recipient's own antigen-presenting cells act as scouts, infiltrating the new organ, capturing fragments of its foreign HLA proteins and transporting this enemy hardware back to the command centers, the lymph nodes, for analysis. This briefing activates a new wave of T cells specifically programmed to hunt down and destroy any cell bearing that foreign signature. Once the organ is identified as hostile, the agency launches its assault. The nature and timing of the attack define the type of rejection. The takedown, hyperacute rejection. This is a preemptive strike, an ambush that occurs within minutes to hours. This scenario unfolds only if the recipient's agency has a pre-existing be-on-the-lookout alert for the donor's antigens, usually in the form of pre-formed antibodies from a prior blood transfusion, pregnancy, or transplant. The moment the new organ is connected and its blood supply is restored, these antibodies swarm the vascular endothelium of the graft. They trigger a massive, instantaneous activation of the complement system's demolition cascade. The result is catastrophic thrombosis and ischemic necrosis. The organ's blood vessels are clogged, its tissue starved of oxygen, and it dies on the operating table. Today, due to rigorous pre-mission screening known as cross-matching, this immediate takedown is a rare event. The primary assault, acute rejection. This is the main battle, the agency's primary coordinated response, typically launched within days to weeks of the invasion. The attack is two-pronged. The first is acute cellular rejection, the ground war. Alloreactive T cells, having been activated by the direct or indirect pathways, infiltrate the organ. CD8+ cytotoxic T cells act as assassins, directly engaging and killing the foreign graft cells. Simultaneously, CD4+ helper T cells orchestrate an inflammatory siege, releasing cytokine signals that recruit a destructive army of macrophages. The second prong is acute humoral rejection, the naval blockade. These cells are activated to produce new waves of antibodies specifically targeting the donor's HLA antigens on the graft's blood vessels. This attack on the organ's supply lines causes inflammation of the vessels, or vasculitis, further damaging the foreign tissue. The long siege, chronic rejection. Even if the initial assault is suppressed, the war is often not over. Chronic rejection is a war of attrition, an insidious conflict that unfolds over months or years and is the leading cause of an invasion's ultimate failure. This is not a dramatic battle, but a slow strangulation. It is believed that T cells, persistently stimulated by the foreign organ, release a slow, steady drip of cytokines. These signals encourage the smooth muscle cells of the graft's arteries to proliferate, slowly and relentlessly thickening the vessel walls. This process, known as graft arteriosclerosis, gradually chokes off the organ's blood supply. It is a slow, ischemic death, a testament to the agency's unforgiving memory. To prevent these outcomes, a desperate strategy is employed: immunosuppression. To save the foreign organ, we must deliberately sabotage our own defense agency. Drugs are used to disarm T cells, to block their activation signals, and to reduce the resulting inflammation. But this counter-espionage comes at a terrible price. An agent that is suppressed from attacking an invader is also suppressed from attacking a microbe or a cancer cell. The patient is left in a state of perpetual vulnerability, trading the certainty of rejection for the constant risk of opportunistic infection. We have now analyzed the agency's ruthless efficiency in identifying and destroying a foreign graft and the dangerous countermeasures required to stop it. This raises a new question: what happens when the agency is not just suppressed, but is fundamentally broken from the start, or systematically dismantled by a master saboteur? In our next and most critical briefing, Part 5, The Agency Compromised, we will investigate the chilling world of immunodeficiency diseases, culminating in the case of the ultimate cellular hijacker, HIV. An agency is only as strong as its weakest link. So far, we have investigated an agency operating at full strength, sometimes with terrifying overzealousness. But we now turn to a far more dangerous scenario: an agency that is compromised, weakened, or has gone completely dark. This is the state of immunodeficiency. The primary threat is no longer a civil war, but a profound and catastrophic vulnerability. The body becomes a defenseless state, where even the most minor microbial intruder can become a deadly occupying force. These agency blackouts fall into two major categories. The first are primary immunodeficiencies, congenital defects where the agency was sabotaged on the assembly line. The genetic blueprints themselves are flawed, resulting in operatives or weapon systems that were never properly built. The second are secondary immunodeficiencies, where a fully functional agency is crippled by an external event or a master saboteur later in life. Our investigation begins with the blueprints, the congenital flaws that compromise the agency from birth. Case files: primary immunodeficiencies, sabotage on the assembly line. These cases are often detected in the first few months or years of life, typically when the initial protection provided by maternal antibodies fades, unmasking the infant's own defective defense system. The investigation into each case is a forensic exercise in genetics, tracing the fault back to a single, broken part in the biological schematics. The defects can occur in either of the agency's main divisions. Sometimes the flaw is in the innate system's first responders. In a chilling condition known as leukocyte adhesion deficiency, the agency's frontline soldiers have a mobilization failure. They are produced, but they lack the molecular grappling hooks, the integrins and selectin ligands needed to grab onto blood vessel walls and exit the circulation. They are trapped in their barracks, unable to reach the battlefield in the tissues, leaving the body to be ravaged by recurrent bacterial infections. In another case, chronic granulomatous disease, the operatives can reach the battle and engulf the enemy, but their primary weapon, a chemical grenade known as the oxidative burst, fails to detonate. The gene for the phagocyte oxidase enzyme is broken. The agents can only contain the invaders by walling them off, forming abscess-like granulomas, but they cannot truly eliminate them. More devastating are the defects that cripple the agency's elite adaptive division. The most catastrophic of these is severe combined immunodeficiency, or SID. This is not a single broken part, but a total command and control failure. Both the T-cell Special Operations Force and the B-cell Long-Range Weapons Program are non-functional. The infant is left utterly defenseless. The most common form is X-linked SID, caused by a mutation in a gene for a cytokine receptor subunit called the common gamma chain. This component acts as a universal communication antenna for multiple signals, essential for lymphocyte development, particularly the IL-7 signal needed for T-cell recruits to mature. Without this antenna, the T-cell division is never built. Another form of SID is caused by a deficiency in the enzyme adenosine deaminase, or ADA. Its absence leads to the buildup of a toxic metabolic waste product that poisons lymphocyte precursors as they develop, effectively shutting down the academies. A more specific, but still deadly, defect is seen in X-linked agammaglobulinemia. Here, the flaw is in the B-cell division's weapons program. T-cell operatives are functional, but the B-cell assembly line is shut down at an early stage. The culprit is a mutation in Bruton tyrosine kinase, or BTK, a critical ignition switch that relays the signal for a B-cell recruit to mature. Without the BTK signal, B-cells never complete their training. The body has no plasma cells and therefore no antibodies. It is left defenseless against a host of extracellular bacteria and viruses. In another bizarre case, Di-George syndrome, the problem is not a broken part, but a missing facility. A large deletion on chromosome 2211 results in the failure to construct the thymus, the exclusive, highly secure academy where T cells are trained and tested for loyalty. Without the academy, there can be no functional T cell operatives. Finally, we have the case of hyper-IgM syndrome. This is a weapons upgrade failure. The B cells are produced and they can manufacture the basic, default ammunition IgM antibodies. But they are incapable of executing the critical class switch required to produce the more advanced, high-affinity Ig-G, Ig-A and Ig-E warheads. The error usually lies not in the B cell itself, but in its T cell commander. The helper T cell is missing the gene for the CD40 ligand, the molecular key that delivers the top-secret authorization code for the B cell to upgrade its arsenal. Without this authorization, the body is stuck fighting with primitive weapons, unable to mount a sophisticated, high-affinity defense. We have now investigated the schematics of congenital failure, the genetic defects that sabotage the immune agency from birth, crippling its first responders or its elite adaptive divisions. These cases reveal the critical importance of every single component in the chain of command. But what happens when the agency is not born broken, but is instead infiltrated and systematically dismantled by the most sophisticated saboteur ever encountered? In our final, concluding transmission for this part, we will open the file on the acquired immunodeficiency syndrome and the sinister mastermind behind it, HIV. We now continue our investigation into Part 5, the agency compromised. We will now open the final and most devastating case file in this section, the acquired immunodeficiency syndrome and the sinister mastermind behind it, the human immunodeficiency virus, or HIV. Narrator: this is the story of the perfect saboteur, a hostile agent so sophisticated, so insidious, that it does not simply evade the immune system. It infiltrates it, subverts it, and turns the agency's own headquarters into a factory for its own replication. The result is not a disease, but a slow, systematic dismantling of the entire defense network. The outcome is the acquired immunodeficiency syndrome, or AIDS, a state of profound vulnerability that has become one of the most devastating pandemics in human history. The scale of the infiltration is staggering. By 2016, HIV had infected 60 million people worldwide, and nearly 30 million have died. The perpetrator, HIV, is a retrovirus, a member of the lentivirus family, a class of slow-acting agents known for their stealth and persistence. It is not a brute force attacker, it is a master of cellular espionage. Its primary target is the very heart of the adaptive immune division, the command and control center of the entire agency, the CD4+ helper T lymphocyte. By targeting the commander, HIV ensures that the entire chain of command will eventually collapse. The infiltration protocol begins with a deadly handshake. The virus particle is studded with a glycoprotein, GPON120, which serves as its master key. This key is designed to fit a specific lock on the surface of our immune cells, the CD4 molecule. But this is a two-factor authentication system. Binding to CD4 is not enough. To gain entry, the virus must also engage a second lock, a deadbolt known as a chemokine coreceptor, typically CCR5 or CXCR4. This dual receptor requirement explains the virus's chilling specificity for the agency's command cells. Once both locks are engaged, the viral envelope fuses with the cell membrane and the virus injects its core into the cell's cytoplasm. Here, it unleashes its most sinister weapon, an enzyme called reverse transcriptase. This is a forbidden technology in our biological world. It allows the virus to rewrite its own RNA genetic code into the language of our cells, double-stranded DNA. This newly synthesized viral DNA, now called a provirus, is transported to the cell's nucleus. Using another enzyme, integrase, it splices itself directly into the host cell's own genome. The infiltration is complete. The virus is no longer a foreign entity. It is now a permanent part of the cell's own blueprint, a sleeper agent hiding in plain sight, waiting for an activation signal. And this is the genius of its malevolent design. The virus remains silent, its genetic code dormant, within a resting T cell. The very signal that is supposed to awaken that T cell to fight an infection, the activation of transcription factors like NF-kappa B, is the exact same signal that awakens the viral sleeper agent. The long terminal repeats that flank the viral genome contain binding sites for these factors. So when the body sounds the alarm to fight off a common cold or a minor infection, it unwittingly gives the order for HIV to begin replication. It is a perfect biological catch-22. The agency's attempt to defend itself becomes the trigger for its own destruction. The collapse of the agency is a brutal war of attrition. The newly awakened virus turns the CD4 plus T cell into a virus factory, and the process of budding new viral particles ultimately kills the host cell. Every day, up to 2 billion new CD4 plus T cells are destroyed, and 100 billion new viral particles are released to continue the assault. The agency's headquarters, the lymph nodes and spleen, become the primary battlegrounds, teeming with viral replication. The very architecture of these command centers is slowly eroded and destroyed. For a time, the body can replace the fallen soldiers, but it cannot keep up. The system is being hollowed out from the inside. The timeline of this collapse typically unfolds in 3 acts. Act 1, the acute phase. This is the initial breach, beginning in the mucosal tissues, where the virus first establishes a beachhead. This is followed by a burst of high-level viral replication and dissemination throughout the body, often accompanied by a nonspecific, flu-like illness called the acute retroviral syndrome. The agency mounts a defense, and HIV-specific killer T cells fight back, partially containing the virus. The viral load in the blood drops to a lower, more stable level known as the viral set point, a grim predictor of how long the agency can hold out. Act 2, the chronic phase. This is the long, silent war, a period of clinical latency that can last for an average of 7 to 10 years. The patient may feel perfectly healthy, but within the lymphoid tissues, a relentless battle rages. Viral replication continues unabated, and the CD4 plus T cell count begins its slow, inexorable decline. Act 3, AIDS. This is the breaking point. The CD4 plus T cell count falls below a critical threshold, typically 200 cells per microliter. The agency's command and control structure collapses. The system goes dark. The body is now a defenseless state, and the gate is open for the devastating opportunistic infections and secondary neoplasms that define the clinical syndrome of AIDS. We have now laid bare the sinister methodology of HIV, a master saboteur that uses the immune system's own protocols of activation and communication to bring about its total collapse. We have followed the timeline from silent infiltration to the final, catastrophic system failure known as AIDS. In the final briefing of our investigation, we will examine the aftermath of this collapse, the rogues gallery of opportunistic invaders and malignant conspiracies that thrive in a defenseless body, and we will close the book with the strange, silent threat of amyloidosis. When the command and control center of the immune agency finally goes dark, the body becomes a failed state, a lawless territory overrun by opportunists and conspirators that were once held in check. The clinical syndrome of AIDS is the chronicle of this collapse, a grim catalog of the infections and malignancies that thrive in a defenseless host. The first to emerge from the shadows are the opportunistic infections. This is a rogues gallery of microbial threats, many of which are latent, sleeper agents that have been living harmlessly within us, suppressed by a vigilant immune system. With the CD4+ commanders eliminated, they awaken and wreak havoc. Pneumocystis gyroceae, a fungus that lives silently in the lungs of millions, emerges to cause a deadly, suffocating pneumonia, often the first overt sign that the agency has fallen. Candidiasis, the agent of thrush, spreads unchecked through the mouth and esophagus. Cytomegalovirus, another sleeper virus, reactivates to attack the retina, threatening blindness, and the gut. In the brain, the final bastion, invaders like the protozoan Toxoplasma gondii establish abscesses, while the fungus Cryptococcus causes a devastating meningitis. With the agency's counter-terrorism division offline, malignant conspiracies begin to form. Cancers driven by oncogenic viruses, which are normally suppressed by T-cell surveillance, now proliferate without restraint. Kaposi sarcoma, a rare vascular tumor, becomes common. It is driven by the human herpesvirus -8, or HHV8, a rogue programmer that forces endothelial and spindle cells into uncontrolled growth, creating the disease's characteristic purple lesions. But the most frequent malignancies are the B-cell lymphomas. With the T-cell commanders gone, B cells that have been infected with another oncogenic virus, Epstein-Barr virus (EBV), are now free from their T-cell handlers. They begin to proliferate uncontrollably. In this state of chronic activation, they are vulnerable to acquiring cancer-causing mutations, leading to the emergence of aggressive lymphomas that often arise in protected, extranodal sites like the brain. The collapse is total. The body, stripped of its defenders, is overwhelmed by a multi-front assault from microbes and malignancies that it no longer has the capacity to fight. Part 6. The silent accumulation, the case of amyloidosis. Our investigation concludes with a final, cryptic case. This is not a story of a battle, an infection, or a civil war. It is a story of a slow, silent, and insidious accumulation. It is the mystery of amyloidosis. This is a disorder caused by corrupted evidence. It is characterized by the extracellular deposition of misfolded proteins that aggregate to form insoluble fibrils, a substance known as amyloid. This material is like a biological ghost. It cannot be removed by the body's cleanup crews, and it does not provoke an inflammatory response. Instead, it builds up silently and relentlessly in the spaces between cells. When stained with Congo red and viewed under polarized light, it emits an eerie apple green birefringence, its unique spectral signature. The investigation has revealed that this corrupted evidence can come from several different sources. In primary amyloidosis, the culprit is a rogue agent. A single, cancerous clone of a plasma cell, as in multiple myeloma, produces vast quantities of a defective, misfolded immunoglobulin light chain. These fragments, known as AL amyloid, are the building blocks of the deposits. In reactive amyloidosis, the substance is a byproduct of a chronic war. In states of long-standing inflammation, such as rheumatoid arthritis, the liver is commanded to produce large amounts of a protein called SAA. In some individuals, this protein is not properly degraded, and its fragments deposit as AA amyloid. Whatever its source, the accumulation of this material is devastating. The deposits are always extracellular, and, as they grow, they encroach on healthy cells, exerting pressure and slowly suffocating them, causing them to atrophy and die. The crime scenes are varied. In the kidneys, the amyloid clogs the delicate glomerular filters, causing massive protein leakage and, ultimately, renal failure. In the heart, the fibrils infiltrate the myocardium, making the muscle stiff and rigid. This leads to a restrictive cardiomyopathy and fatal arrhythmias. In organs like the spleen and liver, the accumulation can be so vast that it causes massive enlargement, giving the tissues a strange, waxy, and firm consistency. The diagnosis depends on finding this ghostly substance in the tissues, confirming its spectral signature with a polarizing microscope. The prognosis is often grim. Amyloidosis is a disease of irreversible accumulation, a slow and silent process where the very substance of the body is replaced by the misfolded ghosts of its own proteins. We have now concluded our investigation into the diseases of the immune system. We have journeyed through the intricate design of the agency and its operatives. We have witnessed its terrifying malfunctions in hypersensitivity, its self-destructive betrayal in autoimmunity, its ruthless efficiency against foreign invaders, and its catastrophic collapse in immunodeficiency. We end with the silent mystery of amyloidosis. The evidence is clear: the immune system is a story of profound duality, a force of exquisite and life-giving protection that holds within it the potential for our own destruction. It is our greatest guardian and, potentially, our most formidable foe.